Professor? Professor?
Yes, I can hardly hear you. Can you put your volume up a bit?
Yeah. Can you hear me better?
Yeah, that's okay.
Hi, good afternoon.
How are you?
I'm good. How are you?
Fine, thanks.
How many questions, I'm sorry, how many more tests do we have this semester? Because on the syllabus it says 4, but on our calendar it only said the 2.
Yes, I know. I left the one out. I'm going to send an email out. You've got one more during the semester. I think it's November 23rd, something like that. You've got one more during the semester. And then at the final test is actually 2 parts. One is like an ordinary test, which makes the 4th test that you've done. And then the other part of the final is comprehensive.
Okay.
So one of those 4 grades, one of those 4 test grades will be dropped. The lowest one will be dropped.
Okay. Thank you.
All right. All right. Thanks for reminding me. I'll make a note and I'm gonna send that email out to tell people. Okay. Okay, good afternoon, everybody, and thank you to all of those who've signed in. It's nice to know that I'm talking to real people. It makes a difference, even if you're not sitting in front of me, which would be much nicer. Okay, so Today I'm going to try and finish this little very, very brief introduction to biotechnology and how in biotechnology we utilize aspects, natural aspects, and manipulated aspects of bacterial genetics. And so I just, to remind you, the one very important thing that I had introduced, the idea of using these little small circular pieces of DNA, these little tiny, they're like tiny chromosomes, the plasmids, which many bacteria have, and which we can extract quite easily from bacteria. We can also construct them. We can make the DNA. There are various ways to arrive at a workable plasmid. And a plasmid which we use to move specific kinds of DNA from one cell to another, we refer to as a vector. But it's important to remember that the plasmids are a natural occurrence, and they naturally pass from bacterium to bacterium. It just so happens that this provides us with a very useful mechanism in biotechnology for deliberately inserting a particular gene into a bacterium. So PUC19, a very, very famous plasmid, based actually on a natural plasmid, which has been highly modified. But there is a couple of important things to note. And that is PUC19 can be inserted into cells using a completely natural system of uptake called competence. Competence arises when a cell is able to take up foreign DNA from its environment and change its genome. By doing that we refer to as transformation. So we can transform bacteria with PUC19 using quite simple systems based on the natural procedures that we see in nature. PUC19 has a number of very important things about it. First of all, it behaves just like a little chromosome. It has its own origin of replication there. So it will reproduce inside a cell that it is inserted into. The second thing is it carries various genes, and one of its particular interest and characteristic for PUC19 is that it has this ampicillin resistance gene. So this gene inserted into any bacterium will confer a resistance to the antibiotic ampicillin. That's first a really useful trait, because this means that if we insert this into a bacterium that is not naturally resistant to ampicillin, we can use an ampicillin plate to select bacteria that have successfully taken up this plasmid. It's a very good way of selecting bacteria that have been successfully transformed. Give them a gene like ampicillin resistance that they do not normally have, and then test them, see whether they have acquired that trait. And remembering that this is true in nature as well. Many of the plasmids that are naturally passed amongst the population will carry genes for things like resistance to antibiotics, heavy metals, all sorts of things. So that is the one thing. We have a very useful gene to select there. But here is another gene, and this is the LAC-Z gene. And this is the gene, a gene which allows bacteria to ferment lactose. And under normal circumstances, that'd be a useful thing to acquire. We've put it there for another reason, though, and that will become clear a little bit later on, because we have to remember that we're using our plasmid as a vector. We're using it to move DNA of our intention into bacteria. And to do so, we make use of any one of these sites here. These are called restriction sites. This is Hindi3, BAM H1, and this is ECHO R1. Those are just names of restriction enzymes. And the restriction enzymes are enzymes which are produced naturally in different bacteria. And their intention is to degrade viral DNA. They do it in a particular way. They only attack the DNA at very specific DNA sequence. And they attack in a particular way that produces like a step. We'll see it again in a minute, which gives us a very useful way of inserting DNA of our volition into this vector. Because if we have a strand of DNA with a gene on it, and we cut that gene on either side with Hindi3, and we then cut our plasmid with Hindi3, we can glue our DNA piece that we've cut out, we can glue it in here at the Hindi3 site. And that does something really particular to this gene here. It actually disrupts it. This gene will no longer operate. The LAC-Z gene will no longer operate if we successfully put our DNA of choice into the plasmid. So we're going to see how that becomes very useful for us in a minute. I just remind you, this is how these restriction enzymes work. Each restriction enzyme has its own particular sequence, DNA sequence that it will cut. GAATTC, I believe, is echo R1, but you can see here. Here's the native DNA. We are interested in this gene here. Let's say we want to insert that gene into a plasmid like PUC90. Here we cut with a restriction enzyme, and it cuts here. You'll notice the sequence GAATTC, and on the other side GAATTC. This we call a palindrome, a palindromic sequence. And because it's a palindrome, the enzyme cuts like this. It cuts as a step in the DNA, and it leaves these ends here, which we call sticky ends. And you can see here, we've got single bases, A, T, T, and there again, A, T, T. We've got those bases. They are desperately looking for their matching bases, We do it in such a way that they don't automatically, we can experimentally ensure that they don't stick back here. And we end up with this fragment here. And here it is here, our useful gene, whatever we were interested in there. And here's the sticky end, this sticky end, AATT. Here's that sticky end, AATT. And we now go to our plasmid, and we come back here. That is using this site here, echo R1. And we're going to cut here with echo R1. And we're going to end up with identical sticky ends, but they're palindromes. And so this AATTC is going to find TTAA end to stick to on our plasmid. Same on the other side. And we're going to end up inserting that gene of choice into this site here, simply because we cut both with exactly the same restriction enzyme. So that's very useful. This isn't actually, this took, of course, a huge, long time of research to develop. It's now a very simple procedure, very, very simple procedure. And unfortunately, we're not in lab. Otherwise, we could do some very simple experiments to show you how we can use a plasmid, cut it, put in DNA of our choice, and get it into bacteria, and there, lo and behold, have it work just like as well as any other gene that the prokaryote had. We actually here do use a couple of other enzymes. We use ligase, which you may remember from DNA replication, to ensure that the whole DNA strand is all joined up together correctly. And when we've finished, here it is here. Here's the one sticky end, TTAA, and it's joined on the plasmids, TTAA. Same on the other side here. And there we've inserted our gene of choice into the plasmid. And off it goes to do its stuff. So I just want to remind you that what we have been talking about, all of the things we've been talking about, have been ways in which DNA is naturally passed from one bacterial cell to another. These processes happen in nature. The cells will take up DNA free from the environment. They will take up DNA from other bacteria that conjugate with them. And they will also take up DNA sometimes when they're infected with viruses, because which is a process called transduction. And that was, I finished off last time describing transduction to you. I'll just quickly tell you again that transduction is a system where viruses attack a bacterial cell, as the normal process for phages, for bacterial viruses, is that they, at one point or another, they will degrade the genome of the host. And they stop the host from working properly, and instead they co-opt all of the machinery of the cell to start to manufacture viral particles. So these are all proteins with little cases with all the legs and everything. It's all viral protein, and it's manufactured by the bacterial cell under direction from the viral genes. But at the end of the whole process, these little packages, if you take these packages and you put them together with DNA, any DNA of an appropriate size, they will actually package the DNA into the head. Under natural circumstances, when these viruses infect these bacteria, they break up the genome into all these fragments. And at the end, the viral particles take up all the DNA that is supposed to be viral DNA. But every now and again, they'll pick up one of these fragments of bacterial DNA maybe with useful genes on it. And they'll go along to another cell and they'll inject that DNA into the cell. This is the natural way that transduction moves genes around the population. But it also offers us an opportunity for experimentally moving DNA from one cell to another. Because what we can do is we can manipulate this whole system in various ways. We can either do it in vivo by causing bacteria to be infected with viruses that will then take up DNA that we want them to take up inside the cell. We can extract the viruses then and use them to, like little injection syringes, to inject that DNA into our cells of choice. So this is a natural system, but we can manipulate it. I won't go into all its various experimental ways that we can do it, but phage transduction is a very useful way in biotechnology for inserting large pieces of DNA into bacterial cells. So let me just go back here to this slide here and point something out. I've said here that we have extracted a gene of interest. We've got a gene somewhere in here that we are interested in, that we want to move into bacteria. Well, how do we get this DNA? How do we get enough of this DNA to make it useful? We actually need quite a lot. The techniques that we use are not by any means 100% efficient. And in fact, we sometimes need quite a lot of the DNA in order to make it, to be able to usefully manipulate it experimentally. So for many years, in the early days of biotechnology, all they did was they just grew up masses and masses and masses of bacteria and then extracted the DNA and then laboriously cut it and purified these fragments for insertion into the plasmids. You can certainly do that. It is one way of getting enough of your DNA of choice to work with. But nowadays, there is another way. And it's a set of reactions that we run called the polymerase chain reaction. And it is a way of taking small amounts of DNA and amplifying them. Amplification means that you increase the number of copies of that DNA. And the PCR chain reaction is actually, in theory, a very, very simple process. But it offers us one very key, useful attribute, which I'll point out to you in a minute. And that is we can really home in on a genome to exactly the gene that we need to amplify. We don't have to go through all of that business of cutting it and everything. We can actually get the PCR reaction to recognize a specific area of DNA that we're interested in, and then just amplify that over and over and over and over and over again until we've got a rich solution of that particular DNA fragment. This kind of amplification can also be used, of course, for all sorts of other things as well. We use it in examining and identifying disease processes. It's used forensically. That is probably where you've heard of it as well. It's used forensically to amplify DNA that has been left at a crime scene for identification and all of these sorts of things. There are various twists to the polymerase chain reaction that can be done. For example, we can even use, instead of using DNA as a template, we can use mRNA as a template. And I'll describe one particular instance where we do that sort of thing at the very end. Okay, so here's the theory behind the polymerase chain reaction. First of all, I should explain to you that it utilizes a DNA polymerase, which is very, very specialized. It's a DNA polymerase which is extracted from a bacterium that lives in hot water springs. Now, under normal circumstances, DNA polymerase, which is an enzyme, it's a protein, is most DNA polymerases are very, very sensitive to temperature, and they denature at higher temperatures. If you take them up, most DNA polymerase, normal DNA polymerase, you take it up above about 45, 50, it starts to denature. But we're going to use a special DNA polymerase, and that is, it's extracted from a bacterium called Thermus aquaticus. Sometimes you'll see it as Thermophilus aquaticus. But that TAQ, TAC, and this is called TAC polymerase. If anybody says to you, I'm using TAC polymerase, you immediately know this is a polymerase which is resistant to high temperatures. In fact, it's very resistant to high temperatures, and it works in the 70-degree range, but it will survive way above that because it's extracted from this bacterium which is specially adapted to hot water. So that's the first thing. The second thing I need to tell you is about DNA itself. You'll remember DNA is the double strand, and then between the strands we have the bases matched A to T, C to G. They are matched with hydrogen bonds. Those hydrogen bonds are quite weak, but the backbone, the sugar phosphate backbone, is very strong. We can separate those strands from one another very easily simply by heating them. If we heat up a DNA strand, it will move apart from one another. We call that denaturing. But it's not like the denaturing of proteins, which is irreversible. This is a reversible denaturing, because when we cool the DNA down again, it faithfully gets back together again with enormous fidelity. It's a very useful thing because we can split DNA apart, bring it together, split it apart, bring it together. We can do that over and over and over again. So this is actually the basis of the polymerase chain reaction, because what we can do is we can split our DNA apart like that, and then we can use our Taq polymerase to duplicate the strands, one in one direction, the other in the other direction, and we end up with 2 strands. Now, if we heat those up, split them apart, do the same thing, we end up with 4 strands, 8 strands, 60. Before very long, we've got a gazillion strands from a simple series of heating, cooling, heating, and cooling. So here's the actual procedure. The first thing is, you'll remember that no replication of DNA by polymerase will take place unless there's a little length of RNA primer put down. So the first thing is we need primers. This is very, very useful, because if you can imagine a normal genome with all its miles of DNA, we have to find our gene somewhere in all of that DNA, right? We have to isolate that gene, because we don't want to be replicating the whole genome. We only want to be replicating that small piece of DNA which is useful to us. Our primers give us a way of labeling that, of putting a molecular label on it, because the primers will only stick to very specific DNA. So we first of all, we do have to do the research. We have to know the DNA sequence on either side of the gene that we're interested in. But once we know it, we design primers. We design a messenger RNA strand that will stick there, or stick there and stick down here. And that RNA primer will allow our Taq polymerase to work and to duplicate the strand, that direction and that direction. We first of all, we have to have primers present. Then we have to have the polymerase. And then we have to have lots and lots of these nucleotides, which are going to be used to duplicate these strands. So we use A, T, G, and C, adenine, thymine, guanine, cytosine. And we put them in a form. They actually have, it's a nucleoside triphosphate. That's what it's called. Nucleoside because it's free from everything else. And it has a triphosphate, 3 phosphates on it. That is because this is an active process. It requires an input of biological energy. But just as in normal replication and everything in the cell, the energy for building the nucleic acid strand is derived from those 3 phosphate groups. 2 of them are broken off. 1 is left to make the bond, and the energy is used to forge that bond. Are you all right? Is everybody with me so far?
Yes, Professor, could you just reiterate what the difference is between a nucleotide and a nucleoside again? Yeah,
a nucleotide is what we saw as it was going to be joined. It's a sugar with a phosphate group on and a base. So that's a nucleotide. Nucleoside is as it occurs free in the cytoplasm with 3 phosphate groups on it.
Okay, so it's just whether or not it's actually integrated into the
Correct.
DNA or free.
Correct. Yeah, that's the simplest way to think of it, yes.
Thank you.
Okay, so what do we have? We have DNA, our target DNA. We've got RNA primers, which are only going to stick on either side just of our target DNA, our target gene, or whatever it is. We've got our DNA polymerase, which is going to work at a high temperature. And we've got lots of nucleotides put in the form that they have these 3 phosphates attached to them. That's going to supply them with energy. So let's just have a look at this. We run it through what we refer to as cycles. And so here's our DNA, okay? And we're going to heat it up. We heat it up to 94 degrees Celsius. That's really hot. But we're using a DNA polymerase that is very resistant to heat. It will survive perfectly well. At that temperature, 94 degrees Celsius, this strand of DNA will split apart. Now, you may start off with a lot of DNA because you've used a whole genome, but you put into this solution, you've put your primers, and they will only stick to a very restricted area of the DNA. They will stick here where they match, where they can base pair and make a hybrid. So this is RNA primer, and it sticks to the DNA. To get it to stick, we lower the temperature to about 60 degrees Celsius. And during that time, the primers stick on, and we call that annealing. So the primers stick on. And now we raise the temperature again, and we raise it to 72 degrees Celsius, because a 72 degrees Celsius temperature cues the DNA polymerase to start working. And the DNA polymerase will run and will duplicate that strand, and it will duplicate that strand. And what do we end up with? Well, you know from DNA replication that if we do this, we end up with 2 copies that are identical to one another. There's one, and there's the other, and they are identical. In this simple cycle, 94 degrees, 60 degrees, 72 degrees, denaturing, annealing the primer, and then replication, we end up with 2 strands of DNA where we had one before. Now, all we do the same thing all over again. We run another cycle. We raise the temperature again. We raise it up to 94 degrees Celsius. All of this DNA will split apart. We lower the temperature to 60. Primers will stick on to all of these strands. We raise the temperature again to 72. The DNA polymerase duplicates all of the strands. We end up with 4 strands. Every time we cycle, we double the amount of DNA. And within a short time, we've got millions of copies of the DNA. We usually run, for a start, don't forget, you won't only have one strand, right? You've probably got quite a few of examples of the gene that you're starting off with. But we usually run for 30 cycles. And that provides us usually with a rich solution of DNA. And it's all this short piece of DNA, just what we were interested in, not all the rest of the DNA. It's this, we can easily separate out this DNA which is short and which only contains the gene that we are interested in. And the reason for that is because we knew that our primers would sit on either side of the gene that we were interested in. So that is the basic principle of polymerase chain reaction. I should tell you, first of all, this is done in a PCR machine. It's called a thermal cycler, and you make up your solutions of this, and you put it into the thermal cycler, and you switch it on, and you go away. And you come back when 30 cycles have run, and there you have in your little tube, you'll have a rich solution, hopefully, of, if everything went right, just of the DNA that you were interested in. It's a marvelous process, and it completely revolutionized the science of bacterial genetics, first of all, and second of all, of biotechnology, because it gave us such an easy way for isolating genes of interest and then amplifying them to a point where they could be useful. So let's see if this works. I think that you won't be able to hear the sound for some reason or another. Oh, man.
Professor, I had a question.
Yes, go ahead.
So you said that you can separate the DNA strands through heating and that it's a reversible process. How can you reattach the DNA back together after you've already got your the copies of the gene that you wanted?
Why would you want to do that? I'm not quite understand. Because what you're doing to follow this strand. Okay, well, here's our double strand, and we heat it to separate it, and each half is going to be replicated. So this strand, the single strand there, ends up double-stranded. It's matching half, ends up double-stranded, but these two are identical to one another. And this is all you're interested in. So just for my, purposes of my kind of example, that little piece there. So let's just pretend that this is what we amplified with PCR. Okay, and we got it up to such a high concentration that we could actually safely put it in with plasmid and run the experiment to actually insert it into the plasmid. So I didn't quite understand what you meant about putting it back together again.
Yeah, because you said that when you heat the DNA, that's denaturing, but this is the type of denaturing where it's reversible.
Okay. Yes. Okay, I understand. I understand what you mean. I understand. Sorry, I didn't quite get it. Yes, you can experimentally, if you want to, heat up DNA and separate it, and then cool it, and it will get back together. We don't do that here. Here we're just separating,
okay.
and then we're doubling it up. But if I took DNA and I heated it up, all the bonds, hydrogen bonds would break. They break very easily. And then if I cool it, it gets back together again. It is something that we use in some other techniques, but not in this. So, let's see if I can get this to actually do something. So this is the target DNA. So we're going to double this each round. Here come the primers. This is annealing. Remember, it can only add in this direction and that direction. This is the 3' end. That's the 3' end. There it goes. That's the DNA polymerase. Now we do the same thing again. There's adenine, cytosine, thymine, guanine. Okay, I think that you probably could not hear the commentary, but if you go to the PowerPoints on Canvas, you will be able to hear them on your computer. I don't know why it doesn't work on Zoom, but it doesn't. This is just be quick. This is just taking you through the cycle. So first of all, we denature, and the DNA strands separate. Now we lower it to allow priming, annealing. Here's the primer. Primer only sticks where it matches. (72 degrees), DNA polymerase works. Now we redo it. Over. Watch the temperatures here. So this is the thermocyclase. This is typical, exactly what it looks like. I just want to try and hurry him up a bit. You can watch here as it cycles. You can see the number of copies of the DNA rising, doubling each time. We're only about 5-6 cycles in, and we're going to go to about 30. By which time we usually end up with literally with billions of copies of the gene that we were interested in. Okay, that's enough. Okay, so now I just want to repeat that many of our experimental ways of inserting DNA into a cell with a vector are based on natural systems. So, for example, PAC19, the one that we've been looking at, you can get that into cells like E. coli by so that it will transform the cells. And in order to do that, in actual fact, in nature, E. coli doesn't do that very easily. But experimentally, we can force E. coli to take up. We can do this by various means. We can heat shock E. coli. We can give it an electrical shock. There are various ways of forcing the cell membrane to become porous to DNA. And when we do that, we say that the cell has become competent. Many cells are naturally competent. E. coli in general is not, but we can make it competent experimentally. So there we're taking a natural system and we're manipulating it experimentally to our advantage. So that is a manipulation of the process of transformation. This, giving it an electrical shock is called electroporation. It actually makes the cell membrane leaky. There is another way experimentally that we can get DNA from one cell into another, and that is by growing up cells which we know have the plasmid of interest, for example, and then getting them to artificially fuse with cells which do not. And you can get two cells which are completely different to one another, with two completely different properties. You can get them to fuse and end up sharing their genetic information. This is called protoplast fusion. And then also we can start to take systems like the phage transduction systems. We can also use conjugation systems. We can use all of these ways of inserting DNA, which is of interest to us, into a cell which does not normally have it. But there is a problem. And that is that we have to be able to tell, first of all, whether we are undertaking two experimental procedures. The first experimental procedure is we're taking a vector like this, and we're inserting a gene, which is of interest to us. We're then taking the second part of the experiment is we're taking this vector and we're inserting it into bacteria whose character we know perfectly well and which we can probably grow easily and everything else, like E. coli. We've done two things, and we have to check to see whether or not both of those procedures were successful before we proceed growing our transformed bacteria and hoping that it's going to do what we want it to do. We have to check, first of all, whether we have put our plasmid into the bacterium successfully. That is the utility of this resistance gene here. This ampicillin resistance gene is a marker for us. For example, E. coli is not normally resistant to ampicillin. It won't grow on an ampicillin plate. We make a medium, a solid medium, agar, with ampicillin in it. And bacteria which are sensitive to ampicillin will not grow on it. And under normal circumstances, that would include E. coli. But if we have successfully inserted this plasmid into E. coli, it will grow on the ampicillin plate. So in other words, if we do our whole procedure, do whatever we make the E. coli competent in a test tube, we mix it up with the PUC19 plasmid, hopefully with an insert into the PUC19 over here. We hope that some of the E. coli cells are competent and take up the PUC19 and then go on about their lives. How are we going to find the ones that have the plasmid? Simple. We grow them on ampicillin plates. If they grow, they've got the plasmid. If they don't grow, they're dead. And they're dead because they don't have the plasmid. They aren't resistant. They die if they are not resistant. And this is their only source of resistance, is the plasmid that we put in. So that's one step. That tells us that we've got bacteria with plasmid in. But it doesn't tell us whether those plasmids that the bacteria have taken up have successfully integrated the piece of DNA that we were interested in. So in order to do that, we need some other means. And here's there are many ways of doing this, but I'm just going to tell you this one to illustrate for you the logic behind this kind of selection. First of all, this is a LAC-Z gene. It is a gene which confers the ability not only to break down lactose, but also to break down some substances which look a bit like lactose, which aren't actually lactose. And this is called a beta-galactosidase gene. And it is able to produce enzymes which will break down a sugar called galactose. We actually use a manipulated form of galactose called X-gal. And the procedure is that this gene will produce enzymes which can break down this X-gal substance. And when they do, they produce a bright blue color. Here's the logic. If we grow these bacteria on a plate with ampicillin in it and with X-gal in it, then we should be able to see whether this is working and whether that is working. So let's work it out. We do our whole experiment. We put the plasmids into the bacteria, we hope, and then we grow them on our plates with ampicillin and with X-gal. Well, we get colonies. All of those colonies are clones of bacteria that have this resistance gene. But when we look at the colonies, we find that some of the colonies are white and some are blue. Well, the blue colonies are colonies where this gene, the LAC-Z gene, is operating. and it's breaking down the X-gal and it's laying down blue pigment. But right nearby them, there are some white colonies. And those white colonies are colonies where the LAC-Z gene is no longer operating. This enzyme, beta-galactosidase, is no longer being produced by these bacteria. Why? Because we interrupted the gene. We put in DNA of our choosing, and we destroyed the activity of this gene. So now all we have to do, we plate our experimental bacteria on media with ampicillin and with X-gal, and we pick not the blue, but the white colonies. And we can be pretty sure that amongst those white colonies, there are going to be some with this gene, our gene of interest, inserted entirely into this plasmid. And once we've done that, we can make that part of any bacterium's genome by inserting it into. It's a neat system. We can actually do, I just put this in as a reminder, that we can actually, quite often this is used in a phage transduction system. And we get phages to actually pick up the plasmids and insert them into bacteria. And then we do a blue, what is called a blue-white screening. And it's that exactly what I've just described. So try and get that logic into your minds, because this is a very, very classic selection system. There's just one last thing that I want to mention about all of this recombinant DNA in prokaryotes. As long as we're just using prokaryotes, it's actually much simpler, because the genes are relatively simply structured as single stretches of DNA, which we can extract, and we can move them from bacterium to bacterium. It's a lot more difficult to work with eukaryote DNA. And the reason for that is that you'll remember the DNA is kind of hyphenated. The genes are hyphenated. There are exon segments and intron segments. Exon segments and intron segments. And when those are transcribed, we end up with a messenger RNA transcript, for example, that has these areas that need to be edited out. So, for example, if we go into a eukaryote genome and we start amplifying a particular gene, we amplify all the introns and the exons. And we end up with DNA which is not functional in a prokaryote system. A prokaryote system can't make product from this gene because it doesn't have the machinery. So this is a big difficulty. If we want to be taking eukaryote genes and putting them into bacterial systems to amplify them, grow them, work with them, etc., we have to use another system. And this is what it is. What we do in working with eukaryotes is, instead of extracting the DNA, what we can do is we can extract the mRNA product of a gene. We extract the mRNA product of the gene, and we're going to work with that. And one of the things that we can do is that we can take a substance called reverse transcriptase. Reverse transcriptase is actually the enzyme which is present in the HIV, in human immunodeficiency virus, and it does something very strange. It, instead of having a DNA replication, we're now going to use the mRNA, and we're going to read the mRNA and produce a DNA strand. That's what reverse transcriptase does. Normally, transcription we read the DNA and we produce messenger RNA. This is called reverse transcription because this enzyme will transcribe messenger RNA and it will produce a DNA strand. And that DNA strand we can then go on to use in PCR. And in actual fact, we can include this whole process in the PCR reactions, in which case it's called RT-PCR. And doing that, we can use eukaryote messenger RNA as the template. And we end up with a copy, which we call a cDNA, copy DNA, which is the functional part of the eukaryote gene. But now it's in a form which, in fact, prokaryotes can use. It no longer has the introns, the exons, or anything in, and it is utilized by bacteria just as if it was one of their genes, and we can produce a product from it. For example, the product that I always return to, because it's really easy to understand, is insulin. Much of the insulin which is produced now pharmaceutically is produced from in bacteria, in bacterial bioengineering systems, and its origin was this exact process. They took the messenger RNA from the insulin gene, they did a reverse transcriptase copy of it, and ended up with cDNA, a cDNA copy, which is just like the gene, excepting it's had all the introns cut out and stitched. Okay, you know, I think I've probably just about done enough for your brains for the day. Does anybody have any questions?
I just had one clarifying question.
Sure.
So for eukaryotes, you still use the same processes. Instead, you just use the DNA from the reverse transcriptase, right?
Yes. What you do is you actually don't worry about the DNA. You extract the messenger RNA from the gene. You get the gene to read off masses of copies of messenger RNA, because in processing the messenger RNA in eukaryotes, they naturally remove the introns and stitch all the exons together. So this is the copy that you end up, this is the messenger RNA that you work with here. Okay, so you get the cell to do all of this for you, and then you come in and you scarf this messenger RNA, and you get the reverse transcriptase to give you a cDNA copy of the messenger RNA. And that way you avoid the necessity to do all the cutting yourself.
So essentially, you're just using DNA made from the mRNA.
Correct. Exactly.
Okay.
Yes, exactly.
Professor, I just had a question about the blue-white screening method.
Yeah. Yes.
I understand the part where the ampicillin resistance gene would help with the selectivity because it would not grow otherwise, right? If it didn't
Correct.
take that up. The part that's a little foggy for me is with the blue and the white colonies. Can you tell that it has taken up the plasmid? Because if it had taken up, I guess I'm trying to figure out why the gene of choice would be, how it's inserted into that particular portion.
Yes, okay. That's a perfectly good question. Let's have a look at this diagram here. Remember what I said, you've got to test for two things. The first thing you've got to test for is did the plasmid go in? And that we can see because they grow on ampicillin plates. So that's what the function of this part is. It confers ampicillin resistance. So any cell that is ampicillin resistant and grows, it has the plasmid in it. But that doesn't tell us whether we successfully inserted our gene of interest into the plasmid. Remember, we're using the plasmid as a vector. We're using it as a way of getting our gene, our useful gene, into a bacterium. The ampicillin only tells us that the bacterium took up the plasmid. It doesn't tell us whether or not we successfully inserted the gene that we wanted to go in. So that is the utility of this. This LAC-Z gene works from one end to the other. It actually is transcribed from one end to the other to produce its messenger RNA and then its product, okay? But if we've successfully inserted our own DNA, we interrupt this gene, and the gene can't be transcribed. So what we're doing, we're testing for a negative, okay? If we now plate our bacteria on ampicillin plates with X-gal in them, any bacterium that is still producing LAC-Z will produce blue colonies because it's successfully making LAC-Z. If it's successfully making this product, it means there's no insert here. So that was a fail. It was a success because it took up the plasmid, but the plasmid it took up doesn't have our gene of interest in it. So what we need to look for are the colonies which grow on ampicillin, but which can no longer produce the blue color. They look white. So we pick the white colonies, and for sure, some of those bacteria will have the whole of our gene inserted here into the LAC-Z gene, which totally disrupts the LAC-Z function, and that's why they are white.
Okay, that makes sense. And is the gene of choice, quote unquote, like attracted to insert there because of those particular restriction sites that are in the plasmid? Okay.
Exactly. It goes in there because it shares those sticky ends. It shares those sticky ends because we cut it with exactly the same restriction enzyme.
Got it. Thank you.
Okay.
I'm still confused on that. Don't you want the LAC-Z gene?
No.
You don't want that, but you want the ampicillin.
Yes.
That's so
exactly.
Confusing.
It's like a negative marker. If they are still successfully using this, it means our gene, the gene we were interested in putting into this plasmid, didn't go in. Okay.
Because of the restriction enzymes.
No. Hold on. Let me just go here. Where's our little? There it is. Have a look here. So let's just say, I'll just use insulin as an example. Say we've got an insulin gene sitting here, right? And we cut that insulin gene out. We cut it out by digesting the DNA on either side with this restriction enzyme. And we end up with this fragment. Here's our insulin gene, let's say, sitting here in the middle. And on either side, it's got these sticky ends, AATT, and on the side, AATT. So now we go to our plasmid. And we're going to cut with exactly the same restriction enzyme. And what we'll end up with is this. We end up with the same sticky ends. We'll end up with a TTAA that this could bind to, a TTAA that this could bind to. And it fits there, it fits in there. There's a TTAA there. Here's the plasmid, CTTAA. Match T to A, T to A, A to T, A to T. That's the usefulness of the sticky end, of this palindromic sticky end. It's going to stick, if I digest with exactly the same restriction enzyme, I produce matching ends. Here it is on this side, and here it is on this side. And that means that I can insert my gene of interest into the plasmid. But when I do that, in this case, in the case of PUC19, what I'm doing is I'm doing that insertion here. And I'm doing it into the LAC-Z gene. So that this gene, if I've been successful, and I've successfully put my gene of interest in here somewhere, I will disrupt the LAC-Z gene. So all I've got to do is I've got to test first for ampicillin. Growth on ampicillin means it's got the plasmid. Then I've got to test to see whether the LAC-Z gene has been disrupted. If the LAC-Z gene has been disrupted, it's probably because I successfully inserted my gene of interest in here. So that's why we test for the negative. We test for the loss of function of this gene. That means colonies who don't have this functioning anymore, they are white in color. Colonies which are still successfully producing LAC-Z, they don't have our gene inserted, and they will be blue. So we avoid the blue and we'll pick the white and grow up the white colonies.
Okay, that makes much more sense. So you want the LAC-Z to be disrupted.
Correct.
Okay.
That's our, for us, that's the positive sign. Okay. Not for the LAC-Z might be a bit ticked off about it, but for us, it's the positive sign that we've successfully put our gene in there.
I just wanted to make a quick comment that it's really fun to learn about this right now because it's one of the COVID-19 testing methods is the PCR test.
Exactly. Correct. Correct. And that is exactly what they're doing with the PCR. They are going in with very specialized primers to isolate viral DNA and amplify it. I should say, anyway, never mind. They're going into to amplify viral nucleic acid. It's not actually DNA, but viral nucleic acid, and amplify it. Okay, any others? All right, thanks so much for coming and joining in the live class. I really do enjoy being able to talk to real people. But of course, this class is recorded and it will be posted as well for you to use again if need be. All right. And with that, if you don't have any more questions, anyone? I'll say good night.
Thank you, Professor.
It's a pleasure.
Thank you. Good
Thank
night.
you, Professor. Good night.
Bye.